So the title of my talk is how to single-cell epigenomic tools help us interpret the non-coding disease risk variants. I have a couple of disclosures to make: epigenome technology and enrichment genomics. Sequencing of the human genome was done 20 years ago, and it was a truly historical event because it gave us the roadmap or blueprint of the life. But reading this blueprint has been nothing but challenging and difficult. And this in part is because only 2% of this genome is protein coding and readable by using the genetic code, while the vast majority of the sequence were non-coding. And for a quite long period was thought to be junk. But that notion has been thoroughly refuted in light of recent evidence. And one of the key recognition over the last decade is that non-coding DNA do play a role in development and disease. In particular, sequence variations in this part of the DNA are also recognized as key drivers of human traits and disease. For example, a single nucleotide change in the enhancer known as ZRS is responsible for this traits called polydactyly, where the individual carrying this sequence change somehow develop an extra thumb. Over the years, genome-wide association assay analysis has revealed hundreds of thousands of such DNA changes that are associated and the most likely causal of thousands of different traits and disease. Most importantly, 90% of those changes in DNA occur in the non-coding part of the genome. And that's where the problem began. And that is we don't have a tool to understand the function of such sequences. The primary hypothesis is that such non-coding variants may disrupt transcriptional control elements such as enhancers to cause disease by perturbing gene regulation of target genes in disease-relevant cell types. This all sounds great, and there has been a growing amount of evidence supporting it. But to use this theory to advance our understanding of human disease and development still face several major challenges. First, the maps of regulatory control element is still incomplete, especially we lack the cell type specificity of annotated control element. Secondly, the target genes of most such elements remain to be precisely defined. And thirdly, how each element contribute to gene regulation, how sequence variants disrupt that function is also largely unknown. We and others have taken a new approach to address this challenge, and we call it epigenome sequencing here. This is because we have recognized that changes in DNA methylation state or histomodification here can actually influence gene regulation in a cell type specific manner. And correspondingly, if we have the knowledge of the whereabouts of such epigenetic changes, we could then annotate functional elements in the genome in different cell types. So the last decade have seen a rapid progress of this field because we were able to combine conventional biochemical assays such as CHIP or DNA's hypersensitivity assays with next-gen sequencing technology to really reveal the genome-wide distribution of various histomodification marks or DNA methylation states. And from these earlier studies, we and others have demonstrated that histomodification can be really predictive of cell type specific activities of genes or regulatory elements. For example, in collaboration with National Axel Wissel group from Lawrence Berkeley National Lab, what we discovered was that the stronger the histone isolation signals at the sequence, the more likely this element is going to function as a tissue-specific enhancers during development. This really laid the groundwork for us to use histone modification signals to map sequences that function as enhancers and determine their cell type specificity during development. There is one problem, though, in the conventional assays. They typically take heterogeneous tissues or mixed populations as the input, and the output is a population average of signals, which are crude, imprecise, and potentially missing important signals, such as from the rare cell types. This is what I consider equivalent to the world map in the medieval times, which could be useful, but a lot of times can be misleading. For example, this takes Columbus to find the new America rather than the Indian land. So now we're taking a new approach, drastically improving our ability to interrogate epigenome in the body. We call it single cell epigenome assays. This line of approach take a lot of DNA barcodes to label individual cells, and then sequence many cells all together and resolve the epigenome landscape within each cell based on the DNA barcodes. There are three different approaches that have been developed to add DNA barcodes to the epigenome before library preparation and sequencing. The first one was first developed is a plate-based approach where individual reactions was carried out in each well of a 96 well plates, and this allow you to essentially generate libraries in parallel for hundreds or thousands of cells. A second approach with higher throughput take advantage of the liquid droplet and microfluidics devices such as 10x or that from BioRad. In these cases, DNA barcodes was added to each cell in nano droplet. And the main advantage is that it can be a high throughput, generating libraries corresponding to hundreds of thousands of cells per assay. And due to the commercial devices, this can be carried out in quite painlessly. Third platform, which I'm going to cover quite a bit in the later half of my talk, is called combinatorial indexing. This strategy generated DNA barcodes using a sequential rounds of split and pooling reactions. In each round, you carry out molecular reactions with pool of cells in 96 well plates or 384 well plates. And then you can mix them all together and dispense them into another plate with 96 well or 384 wells. This allows you to essentially create enormous number of combinations of DNA barcodes. This has a very high scalability. We are now able to actually generate millions of DNA barcodes and process millions of cells in parallel using this strategy. And this is why my lab and many others have chosen to use this strategy to study DNA methylome or epigenome in single cell fashion. Initially, a few years ago, we adopted the single cell combinatorial indexing platform first introduced by Jay Shanduri's lab. It's called Sky ATAC. We made improvement of this strategy such that it can be applied to frozen tissues and can also generate robust single cell ATAC-seq data. The idea is that you carry out the first round of molecular reaction by ATAC-seq in 96-well plates and add barcodes using TN5 transposes. And then you mix everything together and then carry out a second round of barcoding by PCR. Then you can mix them everything together, sequence in a single lane. We are able to produce, observe more than 5,000 open chromatin fragment per cell. That was on average, and at very low cost. So far, we have produced close to 1,000 data sets using this platform. One of the challenges dealing with single-cell ATAC-seq data is the sparsity of the data that we see. On average, I say 5,000 fragments per cell. This, of course, can be increased with additional sequencing or different platforms. But still, this is only a fraction of the open chromatin that you expect per cell, given that we're looking at 3 billion bases of the genome. The strategy we took is to first combine cells of the same cell type together by using a clustering approach. And this clustering there are many tools that carry out this clustering. In our lab, we developed this method called SnapHic, in which case we perform normalization and then using diffusion maps embedding to create clusters of the single cells. Each cluster consists of cells of similar cell type, and then we can aggregate them together, identify open chromatin in that cell type, and use that to infer chromatin open landscape in each cell type. So this strategy allows us to then identify open chromatin and candidate regulatory element in the cell. But we also wanted to examine the chromatin states of each element and do in such a fashion so that we can link the dynamic chromatin state to the transcription of the target gene. And in that case, we developed a method called pair-tag. And pair-tag essentially is a method to jointly analyze histone modification and the RNA transcription from the same cell. It was published earlier this year, so I just want to briefly mention this method involves first staining of the cells nuclei with antibodies that against histone modifications. And following that, we add protein ATM5 coupled with DNA barcodes so that we can generate a double-strand break and introduce DNA barcodes to the sites where the primary antibody is located. That allows us to provide the first tag to the histone modification sites. In parallel, for each cell, we also perform reverse transcription reaction with DNA barcodes such that we can generate in the same cell a cDNA molecule. This step is then followed by 2 rounds of a split and pooling reaction, in which case we add a DNA barcode sequentially and then pull and then add together. This created, as I mentioned, a large number of combinations of DNA barcodes. Following this barcoding addition, we then split the DNA into 2 portions, one dedicated to open chromatin generated by the primary antibody and protein A. Second is a library dedicated to cDNA sequencing. So this way we can basically sequence both the histone modification and the DNA and RNA expression from the same cell. Note that this is, in order for us to study tissues such as brain, we actually have to use nuclei as units. Therefore, our strategy is inherently single nucleus combined assay of histone modification and transcriptome. This strategy was first demonstrated using mouse and mouse forebrain and hippocampus. We were able to produce data from 65,000 nuclei corresponding to 4 different 5 different histone marks, lysine 4 monomethylation on histone H3, lysine 4 trimethylation on histone H3, 27 acylation, 27 trimethylation, as well as K9 trimethylation. We carry out first step using RNA read from each cell that allow us to group the cells based upon their expression profiles. And you can see this generated roughly 22 different cell clusters, revealing actually all the known major cell types in these 2 brain parts. The cells from each cluster is then aggregated to give rise to histone modification marks, such as K4 monomethylation, trimethylation, and so on. So this allows us to effectively map the open chromatin, histone modification states of in a cell type specific manner. Another single cell multi-omic assays that my lab introduced earlier was single cell methyl high C. This strategy essentially combines DNA methylation detection using bisulfite sequencing with 3D genome organization detection using high C, single cell high C. The strategy is very streamlined. You perform 3C in individual cells in 384 well plates, and then you perform whole genome bisulfite conversion and amplification, and then add the adapters, DNA barcodes to the library, and then sequence them as a full. This, why we are doing this? This is because using DNA methylation, you can separate cell types very effectively that corresponding to different type of neurons, different non-neuronal cells from, say, from hippocampus. But if you use 3D conformation, this task could be actually quite challenging. So we basically, first step is using DNA methylation to separate this cells, cluster them to group together, and then take the reads to map the 3D genome conformation for that cell, for that clusters of cells. I collaborated with my colleague Wing Hu from Cleveland Clinics to develop a strategy called SnapHic, and this was published a few months ago. What this strategy allows us to do is to take advantage of the single cell nature of the high C map and to call loops. So you can see basically from, this is from a 4,000 single cell high C map generated in Joe Ecker's lab a couple of years ago. Using standard bulk high C analysis, it's very hard to identify loops beyond a few hundred. But now, using from this individual cell clusters, some are as few as 39 cells and others 1,000 cells, we were able to call thousands or tens of thousands of loops from each cell cluster. And this is give us at least 10 or even 100 times higher sensitivity in loop detection than conventional method. So these are the single cell tools that my lab introduced. The next, in the remaining time, I want to demonstrate how we use this type of tools to gain an understanding of gene regulatory landscape in the body, and to use this to interpret non-coding genome. First, I want to briefly go through a recently published study using single cell ATAC-seq to map cell atlas of the mouse cerebrum. In a very productive collaboration, and also as part of the BRAIN initiative funded by NIH, we collaborate with Joe Ecker from Salk and Margaret Barron from Salk to analyze in single cell resolution chromatin accessibility from 800,000 cells from the mouse brain. These cells are largely come from the cortex, olfactory, lobe, and striatum, hippocampus, pallidum from the mouse cerebrum. These are some of the most important parts of the brain because they're responsible for cognitive function and the sensory perception. From this, from this mouse brain, what we did was we take 600 micron slices from frontier to the posterior of the brain. And from each slice, we then, Marga then carve out herself individual regions of the brain using the brain, Allen brain atlas to identify those that correspond to specific neuronal functions. And then we perform single cell ATAC-seq. We carried this out from roughly 100 samples, generating 800,000 single cell profiles. Basically from this set of samples, we then performed clustering, identifying 43 major cell groups and 160 cell types. This includes 71 GABAergic neurons, which are inhibitory neurons, and 68 glutamatergic neurons, type of neurons, which correspond to excitatory neuron. We also identified 21 subtypes of non-neurons shown here, such as microglia cells, oligodendrocytes, oligodendrocyte precursor cells, and astrocytes, so on. What we discovered was that there is enormous heterogeneity of the brain, which is not surprising, but this heterogeneity mainly reflected in the neuronal level. For example, GABAergic and glutamatergic neurons display very high level of regional specificity in each part of the brain. You will see mainly a particular type of inhibitory or excitatory neurons. And if you move to a different parts, such as hippocampus, a different kind of excitatory neurons or inhibitory neurons will be observed. But this is not true for most glial cells, such as astrocyte or oligodendrocyte. They display, by and large, very low specificity across the brain region. However, we do observe a few progenitor cell types to have very strong regional specificity, which reflect our understanding, for example, how neural stem cells are distributed in hippocampus or in other parts. I will go through this very quickly because the paper has been published. We reviewed we actually have published a web portal to for people, for readers to explore the data. This is called Catalyst Mouse Brain. You can basically explore the data that I just showed you. For example, open chromatin landscape for each of the 160 cell types and the peaks that you can identify that correspond to candidate regulatory elements. So by combining open chromatin from 160 cell types, we were able to identify nearly 500,000 candidate regulatory elements in the mouse genome that are active in one or more cell types in the mouse brain. And you can see that the vast majority of such elements are accessible only in a small number of cell types, while a portion of them show very strong accessibility across different cell types. These are mostly promoters as well as insulator elements. The work has now been published, as I mentioned. They contributed to a special issue of Brain Census of Nature 2 weeks ago. So I encourage everyone to take a look at this issue. I'm going to talk about something that haven't been published. It's about how we use this strategy to lay a single cell atlas of regulatory elements for the human body. In this experiments, which it took us 3 full years to complete, we took 30 adult human tissues from up to 4 donors. Total profiled 600,000 single nuclei from these tissues. We perform iterative clustering, identifying a total of 30 major cell groups and 111 cell clusters. This shows how one tissue actually can be decomposed into different cell types. This is human colon. We were able to observe some of the abundant cell types, such as enterocyte and fibroblasts, as well as some of the rare cell types, such as mast cells and tuft cells and Schwann cells. And we were able to then identify open Chromatin landscape for each of the cell types and call open Chromatin using the peak finding algorithm as shown here, and identify candidate control elements for each cell type. So from 111 adult cell types, we were able to map close to 900,000 candidate regulatory control element, and compare this to the previous catalog of so-called candidate control regulatory elements by the ENCODE consortium published last year. We already uncovered more than half of that catalog, but added 400,000 novel elements. And these novel elements actually are mostly found from rare cell types that are not possible to be revealed by bulk assays, which were primarily used by ENCODE. And we also show that this novel class of elements not included in the previous catalog were associated with open Chromatin state in one or more tissues and show strong sequence conservation compared to control sequences. And even compared to unpublished ENCODE reporter assay, we show 13% of this novel class of elements actually have reporter activities. We also, for completeness, included data, single-cell ATAC-seq data published previously by Jay Shanduri's lab from 15 human fetal tissue types. Altogether, we assembled an atlas of 1.3 million single-cell ATAC-seq profiles. We performed using a uniform pipeline to identify cell clusters and generated an atlas of 222 cell types. The red color shows the adult cell types, and the red, blue correspond to fetal. Half of the cell types were identified as adult, and half were identified as a fetal. Many of the adult and fetal cell types show very strong similarity, not surprisingly. For example, macrophage, T lymphocyte, adrenal cortical cells, endothelial cells, and so on. However, many other or more fetal cell type and adult cell types show actually low or no resemblance. Neurons, for example, between fetal stage and adult cells show very low similarity, indicating a very strong developmental regulation of control of gene regulatory programs. Again, by combining open Chromatin landscape of these 222 cell types, we mapped a total of 1.2 million candidate regulatory control elements. And compared to the ENCODE catalog, we uncovered close to 70% of that catalog, and also added close to 600,000 novel likely control elements to that annotation. This is a heat map showing how this the cell type specificity of each of these 1.2 million elements across 222 cell types. And you can see that the vast majority of them are accessible in only a small number of cell types and tissues. And this is consistent with our understanding that enhancers typically are active in a lineage-specific fashion to drive cell type-specific gene regulation programs. So what can we use this map? I think one of the very exciting prospects of using this map is to test this hypothesis that non-coding risk variants of disease and traits may be associated with cell-specific regulatory elements in the body. And if so, we can use this information to predict cell type that are relevant to the disease occurrence, and to even use this information to identify mechanisms of complex trait and disease. Indeed, we carry out an assay called LD score regression assay. This is a statistical approach where you try to sort out the component of the GWAS summary statistics that are most related to a cell type specific regulatory landscape. So we interrogated over 2,000 traits and disease where we have GOS summary statistics, and we were able to find that 240 of them have a strong association with one or more fetal and adult cell types. Shown here in red color are those with strong association with an FDR of say 0.1% or 1% or 10%. Give you a few examples. Allergy, for example, is associated with T cell regulatory landscape. Not very surprising. Atrial fibrillation, a heart condition, is strongly associated with cardiomyocyte. Again, something that we have expected. Type 2 diabetes associated with beta cell gene regulatory landscape, something that also has been shown before. I want to show you something that is very relevant to where we are. So we know that COVID-19 has wreaked havoc in the world. Many people got infected and then they can recover rather asymptomatically. However, a small number of individuals in a population do get severely ill after infection. And we and others have been interested in the question why. Previously, GWAS studies have identified multiple genetic loci where they predispose the individuals for severe COVID-19 symptoms. And this loci are typically found in interferon response genes and also inflammation-related genes. But the cell type related to this process is not known. Using our study, we were able to identify two cell types, alveolar type 1 and alveolar type 2, to be strongly associated with COVID-19 hospitalization cases. What are these two cell types? Well, long and behold, these are two cell types lying the inner layer of the alveolar in the lung. This is where oxygen exchange takes place. This is also where coronavirus SARS-2 is enter the body through its interaction with the ACE2 receptor on top of the alveolar type 2 cell using the spike protein. So this, I think, make perfect sense in that these two cell types are genetically, patients may be predisposed to the infection by having certain gene regulatory signatures among these two cell types. So the next question is, what are the genes and the pathways involved in this process? So we took our single cell atlas map and intersect with fine mapped DNA variants of different traits. We collated 48 disease and traits where we can have fine mapped variants identified. So out of 10,000 such variants, we were able to show that 30% of them are located in the open chromatin in one or more cell types that we have mapped. And this is very strongly enriched compared to expected by chance, which is at least twofold higher than expected by chance. So from this 30%, 3,000 likely causal variants that located in the regulatory elements, we were able to predict their target genes using 3D conformation information. We also were able to predict likely transcription factors perturbed by such variants using a previous published data from SNP-CELEX experiments. So by combining this two lines of information, we actually can predict potential target genes and upstream regulators for 361 likely causal variants. These are listed in a table that we're happy to share with readers. I want to show one cases where atrial fibrillation actually we followed on with deeper interest. Atrial fibrillation is a condition of the heart characterized by irregular heart beating rhythm, as shown here. Regularly, you have about 1 beat per second, but in atrial fibrillation, you will have sometimes fast, sometimes slow heart beating. And the mechanism is not clear. We were able to show that two risk variants of atrial fibrillation are located in a single open chromatin region within the intron of KCNH2. The open chromatin is discovered in atrial or ventricular cardiomyocyte, making this likely enhancer to be specific to the cardiomyocyte. KCNH2, what is it? It is a potassium channel encoding protein. It is basically important for, of course, action potential, right? Because the potassium channel is driving the efflux of potassium ion during the process of action potential. To understand how these two risk variants indeed contribute to electoral property of the cardiomyocytes, we first have to establish that the elements corresponding to the open chromatin indeed are enhancers. We established this using reporter assay in a in vitro differentiated in a cardiomyocyte model done in Neil Chi's lab at UCSD. So using reporter assay, we can show that the alternative allele in the population is able to drive robust reporter activity in this cardiomyocyte. But if you replace the alternative allele with the risk allele, you lower that reporter activity by more than 50%, suggesting indeed that this is a risk variance that is reducing the enhancer activity of this element. We further demonstrate that the enhancer is for KCNH2 because when we perform CRISPR knockout to remove this element, we show that KCNH2 expression is drastically reduced in the cardiomyocyte differentiated from the human embryonic stem cells. We further show that without this enhancer, the cardiomyocytes differentiated in this in vitro system demonstrate takes 3 times long to complete a cycle of action potential shown here compared to the control cells. So this is very significant because action potential is indeed how heart rhythm setting cells is able to set the rhythm. If you have a cardiomyocyte that are unable to respond in the same way as other cells to such a rhythm setting, your electrical property is going to be affected significantly, therefore leading likely to arrhythmia. So in summary, what I've shown is that we generated single-cell atlas of chromatin accessibility for the human genome across 222 fetal and adult cell types. In this atlas, include 1.2 million candidate control elements. We demonstrate how this map can be used to review the disease-relevant cell types and uncover the affected regulatory elements by the risk variants, and using this to understand the mechanism of polygenic human traits and disease. To close, I'd like to thank my colleagues and collaborators who have played an instrumental role in the studies I presented. I mentioned their names during my presentation. I want to specifically mention Center for Epigenomics at UCSD, which is set up 5 years ago as a collaborative center. Its mission is to apply state-of-the-art epigenome technologies to basic science and clinical research. We're set up to collaborate with others so that we do the protocol optimization, we do the production, while researchers all over the world can basically ask the biological questions by collaborating with us, supplying samples, and we provide the data back. Collaborators of the project that I mentioned, Kyle Galton is our close collaborator in the Human Atlas project. Neil Chi also was collaborating on the human heart study. I mentioned others also during this process. I'm particularly grateful for the for NIH, for the Brain Initiative, and for B2B, for the type 2 T2D gene, and other programs that funded our research over the years. I think I have 5 minutes left. I'm happy to take questions. Thank you so much for your attention.
Very nice. Thanks, Bing. That's incredible. If anybody has questions, they can put them in the Q&A chat box here, the bottom of your screen. But maybe just in the meantime, I'll ask a question. First of all, it's so cool that this map can just be reused for any disease, pick out the highest risk SNPs and find the gene. And you sort of showed, the formula for that, but that could be repeated many, many times. And it's really nice to see it get to the functional level. But I was kind of curious about the regional specificity you noticed in the brain project, where the GABAergic neurons and the other, they sort of clustered very differently from each other. But I noticed in the GABA neurons, there was some that had low regional specificity and some that had high. Sorry, you don't have to go all the way back. But I was just wondering if you were able to sort of figure out the genes that would distinguish those two types. Yeah, here. So the sort of the left side of the GABA neurons had low regional specificity and the high. Can you use this data set to sort of now do differential analysis between these two, or are they just so variable that it's not useful?
That's a very good question. So what we can do is perform, of course, differential accessibility assays, which we did in this paper. I didn't have time to discuss that. We also perform a motif analysis to compare what's the driver that are likely responsible for such differences. So we provided a lot of such candidate drivers to look after, and I think future studies to perturb those driver transcription factors will be needed to delineate whether they are involved in regional specificity.
Okay, cool. Okay, so we have some questions rolling in here. From Sarah, we have you been able to distinguish many cell types with this type of analysis? Do we have a sense of how many are distinct cell types there may be?
Yes, Sarah, that's a great question. I have to say, we're only scratching the surface. I would say this is a tip of the iceberg. Our brain study shows that even from a relatively simple organ like brain, you can uncover hundreds of cell types. In fact, using transcriptome analysis, it has now been recognized that maybe thousands of brain cell types exist in this organ. And that is only possible if you actually take a very deep dive, like millions of cells, to be able to resolve that. So for the whole human body atlas, I have to say we're greatly undersampling the complexity of the human body. I think future studies where this scale have to be 2 orders of magnitude for us to reveal some of the rare cell types and also developmentally transient cell state. So I think we're really at the very superficial level.
Okay, then we have another question here from Anjali. Have you looked into the enrichment of somatic SNPs in the brain cells? So I guess comparing somatic versus neuronal, how do you think somatic SNPs could enhance our understanding of germline SNPs?
Well, that's a great question. No, we haven't. I think our study right now is focused on the germline SNPs that were discovered using GWAS. For somatic SNPs, it will require a deeper, more reliable sequencing technology that I think are only become available recently. And we should probably look into that more closely now.
Excellent. All right, well, thank you, Bing. And everybody check out all these resources. I know I'm gonna